Hello, how are you? Welcome to our lesson today. Today, we shall learn about growth and development in plants. And you will realize that the life or the journey of plant life actually begins from the seed. Therefore, growth in plants actually starts from seeds. When seeds mature, they usually germinate to produce now new plants. So we can actually say that growth and development in plants begins with germination in seeds. It begins with germination in seeds. Yeah. Now, before we start or we discuss about growth in plants, I think it's important that we start discussing about how does the seed look like. So we shall discuss about the structure of the seed. Okay, good. Now, the diagram displayed above here actually shows the structure of different seeds. The first diagram, as you shall see here, you will actually work with this particular pointer, the one in red. The first seed actually is a common bean seed. This is an example of dicotyledonous seed. Usually a seed, it is covered by a seed coat. This seed coat here, in most cases, it comprises of two main layers. Yeah, we have the outermost, which is tough, is the tester, while the innermost is usually membranous and transparent in color. It is called yes, it is called the tegmen. It's called the tegment. Okay, tegmen. So we're saying that the seed coat is composed of two main components. We have the tester and the tegmen. Good. Now, so I've said it is composed of the tester and the tegmen. Tester and tegmen. I want to write the word tegmen so that you understand. Yeah. So I've said it is made up of the tester and the tegmen. Say, yeah, tegmen as visible here. So the tegmen usually is transparent in color. So what do you think is the role of the seed coat, which is made up of tester and tegmen? Yes, because it is tough, the seed coat protects the seed from entry of microorganisms. So inside this particular seed, there are also some cotyledons. These cotyledons usually have got a function. What do you think is the function of the cotyledons? The cotyledons usually have got one main function. And the main function here is to store food for the developing seedling. Then we have now embryonic, the embryo. The embryo is made up of the plumule and the radicle. So this embryo usually grows into the seedling. Okay, it grows into the new seedlings. Between the embryonic leaves, and the radicle, we have, and the cotyledons, we have this epicotyle. Very, very important. We shall see it later during germination. And also we have between now the radicle and the cotyledons, we have another part which is called the hypocotyle. These parts are very important, especially during germination. Now, apart from that, we can also have monocotyledons. You can see here that our common bean seed has got two cotyledons. Therefore, this type of seeds is known as dicotyledonous seeds. That is, it is a seed that is composed of two cotyledons. Then we have also the corn seed. Corn seed is the maize seed. We learned under reproduction that the corn actually is a fruit. Yeah, it's a fruit. Now, if you look keenly, the corn seed actually has got an endosperm. The endosperm actually serves what? The purpose of storage of food. So it stores food for the embryo. Okay. Then here we have the cotyledon. It has got only one cotyledon. Okay. Then the embryo comprises of the plumule and the radicle. We have coleorhiza, which is down here. It protects the radicle, and the coleoptile above the plumule. It usually protects the plumule during growth. Okay. So let us go back to the bean seed. Externally, the bean seed, you can see here that usually there is a scar. It is not labeled here, but usually here we have a scar. This scar is a point of attachment between the placenta or the funicle, we shall see later, and the seed. And also above the scar, usually we have the micropyle. The micropyle is a tiny pore up here, which allows water and oxygen to move into the seed. These factors are very important during germination. So let us go to the literature and we continue learning. Good. So we have said that the seeds consist of the seed coat. The seed coat encloses the embryo. Sorry. The seed coat encloses the embryo, and it is composed of an outer covering, which is made up of two layers. The two layers are the testa, which is the outer, and the inner is the tegmen. Okay. The testa is thick, while the tegmen of seed is transparent and membranous. The two layers serve one purpose: to protect the seed from bacteria and fungi and other microorganisms which may damage the seed. Okay, good. Then apart from that, the seed also has got the hilum. The hilum is also known as the scar. This is the point of attachment, a point where the seed was initially attached to the funicle or the placenta while it was still in the embryo or in the embryo. Then we have also a micropyle. The micropyle is just above the scar. Okay, and it is a tiny pore near one end of the seed, and it allows water and air into the embryo. So this micropyle allows air, and also it allows water to get into the embryo. Then we have the cotyledons. We have said that these sometimes are known as seed leaves. They store food for the embryo, and above all, they are either one or two per seed. We said seeds with one cotyledon are said to be monocotyledonous, while those with two cotyledons are called dicotyledonous seeds. Okay. Then we've said also we have the plumule. This is the embryonic shoot. We've said the embryo is composed of the embryonic shoot and the embryonic root. So the embryonic shoot is the plumule, while the embryonic root is the radicle. So radicle is the embryonic root. So the radicle is found opposite to the micropyle. Opposite to the micropyle. Now, we also have seeds that do not store food which actually don't store food in the where in the cotyledons. So seeds that store food in the cotyledons are said to be non-endospermic. What does that mean? They don't have endosperm. Huh? So seeds that store food in cotyledons are called non-endospermic seeds. This one is very common in dicotyledons, while those which store food in endosperm are called to be endospermic seeds. And this one is very common in the monocots. So that actually, with those few remarks, we shall actually stop there. And in our next discussion, we shall learn about the seed dormancy. Otherwise, have a nice time. Let us meet in the next session. Bye bye.